Aim. To understand the structural components and their functions of an autoclave. Apparatus/materials required. Laboratory autoclave, distilled water, thermometer or temperature gauge, pressure gauge, biological media or glassware. Theory. An autoclave is a device used for sterilization of laboratory materials by using steam under pressure. It destroys all forms of microbial life including spores by maintaining high temperature, 121 degrees Celsius, and pressure, 15 psi, for a specific time, usually 15 to 30 minutes. Autoclaving is a vital process in microbiology, biotechnology, and medical laboratories to ensure aseptic conditions. Structural components and their functions. Part. Function. Chamber. The main vessel where materials to be sterilized are placed. Made of stainless steel to withstand high pressure and temperature. Lid/cover. Seals the chamber tightly to prevent steam leakage. Usually equipped with locking bolts and a rubber gasket. Pressure gauge. Displays the internal pressure of the chamber, helping to monitor sterilization conditions. Temperature gauge/thermometer. Indicates the internal temperature of the autoclave. Safety valve. Releases excess pressure automatically to prevent accidents. Steam inlet/boiler unit. Generates and directs steam into the chamber. Air outlet/exhaust valve. Allows trapped air to escape before sterilization begins, ensuring uniform heating. Water reservoir. Holds the water that is converted into steam. Timer/control panel. Allows setting and controlling of sterilization time, temperature, and pressure. Basket/tray. Holds the materials, media, glassware, instruments, inside the chamber. Power supply unit. Provides electrical energy to heat the water and generate steam. Working principle. Sterilization occurs through moist heat, steam, under pressure. When water boils under pressure, it produces steam at a temperature higher than 100 degrees Celsius. This high-temperature steam coagulates and denatures microbial proteins, killing bacteria, viruses, and spores effectively. Procedure. Fill the water reservoir up to the marked level. Load materials to be sterilized into the chamber. Close and lock the lid securely. Switch on the autoclave and set temperature, 121 degrees Celsius, and pressure, 15 psi. Allow it to run for 15 to 30 minutes. After sterilization, let the pressure return to normal before opening the lid. Observation. The autoclave reaches the desired temperature and pressure. After the cycle, materials are free from microbial contamination. Conclusion. The autoclave uses steam under pressure for effective sterilization. Each structural component plays a vital role in maintaining safety, pressure, and temperature during the process.